When it comes to sequencing of peptide, the first thing we have to understand is whether this peptide chain consists of any disulfide bond or not. So, our step 1 should be determining the presence and removal of disulfide bond. So, how come that can be possible? So, removal of disulfide bond can be initiated by two mechanisms. One by means of oxidation, another by means of reduction. When it comes to oxidation, suppose this is our disulfide bond CH2S, this is our C, this is our H, C double bond O, NH3 plus, something like that. S, CH2, C, C double bond O, this is our H, this is our NH3 plus, something like this. So if this disulfide bond needs to be broken by means of oxidation, the second thing that will happen is both these bonds, both these chains will be separated from each other. Both these chains will be separated from each other, giving rise to S minus on this side and S minus on this side. This is by means of oxidation. And when this oxidation takes place, that is conducted with the help of performic acid. Performic acid. On the other hand, when it comes to reduction, this is our disulfide bond. Suppose we are initiating the removal of disulfide bond by means of reduction. Due to reduction, what will happen? These two chains, chain 1 and chain 2, they will be separated from each other, forming sulfur hydryl group on each of the sides. Sulfur hydryl group on each of the sides. So this is removal of disulfide bond by means of reduction. And it can be initiated by two reagents. One, beta markaptoethanol, and second, dithiotherol, DTT, dithiotherol. So first we have to keep this in mind that we need to remove the disulfide bonds. In order to remove the disulfide bond, what we have to do? We have to initiate the process of oxidation either or reduction. If we do oxidation, then that can be conducted with the help of performic acid, and this will give rise to S minus on both ends after the removal of disulfide bond. On the other hand, when it comes to reduction by means of beta markaptoethanol or dithiotherol, what will happen? In each of these chains, cysteine residues will be bearing their sulfur hydryl groups. Cysteine residues will be bearing their sulfur hydryl groups. Okay, so this is our step 1. Coming to our step 2, which is identification of amino terminal end. Identification of amino terminal end can be conducted by three distinct mechanisms. A, by using Sanger's reagent, which is 1-fluoro-dinitrobenzene, 1-fluoro-dinitrobenzene, along with 6 molar HCl, along with 6 molar HCl. But there lies a problem with this technique. As we are applying 6 molar HCl, the entire peptide bond will readily get disrupted instead of determining only the amino terminal end. This is a drawback of this technique. Another thing is we can identify them with dapsil or damsyl chloride. This can also be done. This will help to identify the amino terminal end. But the most effective way to determine our amino terminal end is by using Edman degradation, by using Edman degradation mechanism. So here we will discuss briefly about Edman degradation. Let's see what happens. In Edman degradation, we have our reagent, which is called phenyl isothiocyanate. Phenyl isothiocyanate. Let's see. So this is our PITC structure. Now, suppose there lies our peptide chain. Something like that. There lies our peptide chain, and this is our phenyl isothiocyanate. Now, if we are trying to differentiate the amino terminal amino acid of a peptide chain, then what we have to do at step 1 of Edman degradation, at step 1 of Edman degradation, we have to raise the pH and make it basic. Raise the pH of the solution and make it basic. So what is the purpose of it? When the pH is raised, this NH3 group will lose its H plus ion. This NH3 group will lose its H plus ion. And hence, what will happen? This will form NH2 lone pair with our C R1 H C double bond O, something like this. So this will be formed. Now it has its lone pair, and within the solution we have our phenyl isothiocyanate. So what this lone pair will do, this lone pair will create a nucleophilic attack upon this carbon. This lone pair electron will create a nucleophilic attack on this carbon. As a result, what will happen? There will be our phi n. There will be H. As the H plus has been released from here, that will be received by this nitrogen. We have our C double bond S. And on the other hand of S, on the other hand of C, this entire structure will be accommodated. This entire structure will be accommodated. So this will be formed initially. The structure that is being formed over here, the structure that is being formed over here, this is called our PTC, phenyl thiocarbamoyl adag. Phenyl thiocarbamoyl adag. Within this structure, let's focus on certain things. We have our phenyl isothiocyanate unit along with our first amino acid. So initially, this will later on structure will later on give rise to this structure will later on give rise to a 5 carbon chain structure upon treatment with trifluoroacetic acid. Upon treatment with trifluoroacetic acid. And hence there will be our first amino acid. Now, one thing we need to understand, if this particular peptide chain is known to us, then that won't be an issue. But if this particular peptide chain is unknown and we are initiating a sequencing mechanism, then the first amino acid along with our phenyl isothiocyanate unit, this entire structure needs to be initiated MS, that means mass spectroscopy, so that we can identify which of the amino acid is attached with our phenyl isothiocyanate. So by this mechanism, we can understand or recognize the first terminal amino acid over here. And another thing we have to understand, during the process removal of first amino acid, what will happen? Initially, we have to increase the pH so that the amino terminal H plus can be removed. But later on, over here, we are introducing trifluoroacetic acid. The purpose of it is to again reduce the pH so that our amino acid can easily be removed from the rest of the polypeptide chain. our amino terminal amino acid can easily be removed from rest of the polypeptide chain. So this is the overall mechanism of Edman degradation. But there certainly lies a problem with Edman degradation mechanism. This entire process is conducted within a machine called sequenator. But there is an issue because as per theoretical aspects, the accuracy of Edman degradation is 97%, not 100%. So suppose there lies a sequence lysine, proline, phenylalanine, something like that, so on and so forth. So if Edman, by means of Edman degradation, take a deeper look over here. If by means of Edman degradation, removal of removal of lysine residues has taken place in 97% of polypeptide chain. Then there will be 3% of the polypeptide chain remaining within the solution with amino terminal lysine, and on the second round of Edman degradation, 94%, as 3% accuracy is less, there will be 94% of the polypeptide chain. There will be 94% of polypeptide chain which will bear the proline residue. And rest of the portion, among the rest of the 7%, there will be a percentage of proline with amino terminal end as well as lysine as amino terminal end. So, due to this kind of problems, we cannot state this approach as 100% accurate. So, this is how entire mechanism of amino terminal end determination takes place. Now, after amino terminal end detection, we will now analyze about our step 3, usage of certain enzymes. Usage of certain enzymes and utilizing their properties. Suppose we have, for this purpose, we have mm, trypsin. Trypsin in general, it cleaves the peptide bond. These are all endopeptidases. These are all endopeptidases. So trypsin, what it does, it cleaves peptide bond based upon presence of lysine and arginine at their C terminal end. That means if lysine and arginine are present within that particular peptide chain, then cleaving by trypsin will occur at the carboxyl terminal end. That means the carboxyl group will be provided by either lysine or arginine. I'm repeating myself. The carboxyl group will be provided by either lysine or arginine when it comes to cleaving by means of trypsin. Next, coming to chymotrypsin. In case of chymotrypsin, what is observed? In general, chymotrypsin cleaves the phenylalanine, tyrosine, and tryptophan. Phenylalanine, tyrosine, and tryptophan. Same C terminal end. That means the carboxyl group, if provided by three of these amino acids, then only the cleaving will take place by means of chymotrypsin. Another important enzyme, cyanogen bromide, CNBR. In most of the cases, if there lies a presence of methionine, then it can be cleaved by using CNBR as an agent. Because CNBR can cleave methionine same at their carboxyl terminal end. These are mostly used when it comes to endopeptidases. But there lies certain exceptions also that may cleave at their amino terminal ends too. Like there lies pepsin, there lies thermolysin. Thermolysin can cleave amino terminal end also, specifically for hydrophobic amino acids or bulky group amino acids. Valine for valine, for isoleucine, for leucine. For these cases, thermolysin can cleave at their amino terminal end, not carboxyl terminal end. These are exceptional. Similar goes for pepsin. Pepsin can also cleave at their amino terminal end, so on and so forth. So these are all our endopeptidases. So first point is we need to determine whether there lies any disulfide bond or not. For that, what we have used performic acid for oxidation and beta mercaptoethanol or dithiotherydol for reduction. What they are doing for oxidation, they are giving S minus, and for reduction, they are providing sulfahydryl groups. As simple as that. Based upon this, we can easily differentiate the two chains of the peptide bond. Or if the peptide bond is at all associated with each other in this format, that also can be cleaved by using this kind of agents. Second, coming to amino terminal end determination, that can be initiated by using Sanger's reagent. For Sanger's reagent is 1,4-dinitrobenzene with the application of 6 molar HCl. Dapsil and damn chloride is also there in order to determine the amino terminal end. And lastly, Edman degradation mechanism, which is the most applied aspect of it. In Edman degradation, what we have observed, the reagent is PITC, phenyl isothiocyanate. This is a structure of phenyl isothiocyanate. What it does, in the first step, we have to increase the pH, and as a result, the amino terminal end of our peptide chain will lose the H plus, and as a result, it will form NH2. In its NH2 state, what it has a lone pair electron, and this lone pair electron will readily attack the carbon of PITC, phenyl isothiocyanate. As a result, an entire complex will be formed. This complex is called phenyl thiocarbamyl adduct. And during formation of this entire structure, what will happen? This nitrogen, which has a lone pair, it will create a nucleophilic attack over here and in this carbon, and readily that carbon will again conduct a nucleophilic attack over here. As a result, this NH that is present in its NH form will transform. This NH will transform from NH to NH2 plus. So there will be a partial positive charge over here. There will be a partial positive charge over here. And as a result, this entire structure, this entire first amino acid with the phenyl isothiocyanate unit will be removed by the application of performic acid. So this is the mechanism by which the entire process of amino terminal end determination using Edman degradation takes place. And finally, coming to our endopeptidases like trypsin, like chymotrypsin, like CNBR, which is normally at carboxyl terminal end, but we also have exceptions like thermolysin that lives at amino terminal end of hydrophobic amino acids like leucine, isoleucine, valine, even phenylalanine is there. And there lies pepsin also that can also cleave at amino terminal ends. And lastly, coming to our determination, which is the last step, step 4, determining the carboxyl terminal end or carboxyl terminal amino acid. In order to determine the carboxyl terminal amino acid, we have two distinct aspects. One is usage of reagents like lithium aluminum hydride, reagents like lithium aluminum hydride or hydrazine is there, and there are certain carboxypeptidases. There are certain carboxypeptidases that can determine our carboxyl terminal end. Carboxypeptidases can again be differentiated into three groups. Carboxypeptidase A is there, B is there, C is there. A is for aliphatic amino acid, B is for positively charged amino acid, and C for any of the amino acid. These are the critical things when it comes to determination of C terminal end amino acids. So this is the overall process of sequencing of a typical peptide chain. If you find this video interesting as well as informative, do share with all of your batchmates and comment below if you want solve of PYQs regarding this particular topic, and we will see you in the next video next week. That will be based upon MSMS, another mechanism of sequencing.